I never know if I have to wait for this to load all the way before I can close it out. So I'm going to. Close it. Echo. So when we left off, we were talking about metabolism. Julian said, yes, we are talking about the mitochondria. When we think about metabolism, talking about the area around the mitochondria and then moving into the mitochondria. So that is where we are. We had not talked about that before. Okay. So really what we're thinking about metabolism, we should be thinking about cellular respiration, and you're thinking about the normal stereotypical textbook example of metabolism. What you're thinking about specifically is aerobic cellular respiration. We will talk about other types of metabolism, but that's our real bread and butter. That's our backbone of everything. So we have our equations for that. You don't have to memorize it, but you should know that we're breaking down glucose. That's our carbohydrate here. Using oxygen, that's the part that tells us this is aerobic cellular respiration. We're breathing out carbon dioxide, breathing out some water vapor, and getting a lot of energy for our cells. So we went and we left off, we were at our first step, glycolysis. So glycolysis is where we start to break down the glucose. We can track a lot of different things as we're moving through metabolism. But one of the main things we want to track is what's happening with our big carbon-containing molecule. So we're trying to figure out where all those carbons from the glucose go. And then the other thing we really want to concentrate on is where ATP is being produced, because that's the whole point of it. If we were to pay attention to a third thing, we would pay attention to, in orange, our electron carriers. There would be our NADH. The reason we pay attention to that is really just because we are interested in ATP, and they're going to make a whole bunch of ATP at the end of the process. So it really goes hand in hand with that production of ATP. Okay. So the first thing on this list, right, is that what we're doing with all those carbons, right, we're breaking down glucose, which is a 6-carbon molecule, into 2 pyruvate molecules, which is 3 carbons. So that's what's happening with the carbon-containing molecule. Then we have these branches coming off the sides, right? So in glycolysis, we produce 2 molecules of ATP, and we produce 2 reduced electron carriers with our NADH. We're not going to worry about all the steps that exist in glycolysis. There are 10 of them. There are a bunch of enzymes. You could memorize them if you wanted, but I don't want to, so I don't expect you to. If you need a specific step, that's something you could look up. That would be really a point at which you'd probably be interested in that if you were doing your own research into metabolism. But for our typical purposes of understanding nutrition, understanding how the body functions, we don't really need to worry about each of those steps. So we have a net gain of 2 ATP molecules. The reason this is a net gain is we do have to put in a little ATP at the beginning to kind of jumpstart this process. So if you see 4 ATP at some point, it is true that we made 4 ATP, but we used up 2. So net gain of 2. The number to remember here is 2. Our NAD plus is reduced to NADH. So this is our electron carrier. This is what's helping us carry electrons to our electron transport chain at the end. This is where we're going to get really good bang for our buck with making ATP. So you'll see these terms reduced. You'll see oxidized. We talked about glucose oxidation at some point. So just I want to make sure that we remember, because it's always fiddly when we have our terms. Right? We remember our Leo the lion says grr, right? So when we're talking about losing electrons, that's oxidation. Spell losing. gaining electrons equals reduction. Okay. So oxidation and reduction are paired. These are called redox reactions. We're not going to worry about them for the purposes of this class. But we are going to see these terms, reduce and oxidize. So what's happening here is that as we gain our electrons and become this reduced form of our electron carrier, we can tell it's that reduced form because it kind of tacks on this hydrogen at the same time. So an important thing to know about glycolysis, separate from the fact that we're making 2-ATP and we're making 2-pyruvate, is that there's no oxygen involved in glycolysis. So we are tracking the course of aerobic cellular respiration, which will eventually require oxygen, but it has nothing to do with this first step. And that's important because we're going to see later that glycolysis can happen in low oxygen environments. So we will see that sometimes glycolysis will run to produce ATP without the rest of our subsequent steps happening. So we'll be able to get ATP without using any oxygen through glycolysis. So it's very important to know we don't get here. Other thing to note is that glycolysis is happening in the cytosol. So all the rest of our steps are going to be moving into the mitochondria. But right now we're just floating around in the cytosol in the cytoplasm of the cell. So we're not seeing the organelle yet. And that will kind of link in later to when we talk about muscle cells, because we'll see that how many mitochondria a cell has will have something to do with how much aerobic respiration that requires oxygen is happening in that cell. So if you were only doing glycolysis, you wouldn't really need so many mitochondria. And we'll see that that's a difference between different types of muscle fibers. That's not a for now problem, that's a for later problem. So NAD plus and NADH are these energy transferring nucleotides, these electron carriers. We can technically call them coenzymes because they're going to work in partnership with enzymes in our electron transport chain at the end. So we talked about coenzymes and cofactors as being things that help enzymes work. Basically, the enzymes at the end are going to need NADH in order to function, and without it, they won't be able to produce any energy. So NADH is the reduced form. Reduced means it has an extra electron. So we're always trying to make NADH until we get to the end. Right? We're always trying to pack on electrons until we finally use them up at our final step. So this is our whole flow for glycolysis. These are all the intermediate forms that we are not going to be memorizing right now, but you can see that point at which we're putting in ATP at the very beginning. Right, so here are the 2 ATP that we're putting in. Right, so that's why we had 2 net, because that was 2 in, and then we have 4 out. So there are 2 here coming out, and there are 2 here coming out. So it's a total of 4 ATP, but net 2. So that first part where we use the 2 ATP is the energy investment phase. And then we have our energy harvesting phase. We could break this down into smaller types of phases, but we're not worried about those for now. We're really just focusing on what's going on with ATP, what's going on with the energy. And again, more lovely pictures if you are curious and interested in the chemistry involved. More ATP, more NADH, lots of enzymes. You'll notice we see this word kinase a lot, the common ending for certain types of enzymes, right? We know that those are involved with using up ATP. The last time we talked about some of these words, so I didn't set this up as a question and answer, so just write down your answer for yourself or write down the question for yourself. So how is glycolysis generating ATP? Talks about two types of ways we can make ATP. Glycolysis is one of them specifically. Anyone want to tell me what type of ATP generation they think glycolysis is?
Substrate level phosphorylation.
Exactly. Yeah, it's substrate level phosphorylation. So we're using enzymes to pull phosphates off of another molecule and put them onto our ADP to make ATP. So after glycolysis, we now have these two pyruvate molecules. We need to figure out how to get them into the mitochondria and start breaking those down to do our next steps. So we think of this kind of breakdown of pyruvate as a linking step. Sometimes it's referred to as its own step, but what it's linking is glycolysis with the Krebs cycle. So we're getting ourselves into the mitochondria, so we're transporting. And then we're breaking down pyruvate using an enzyme, pyruvate dehydrogenase. We're producing some of these reduced electron carriers here. We start taking out some of our carbon. So we didn't breathe off any CO2 for glycolysis, but we do start breathing off CO2 for this linking step. What do you notice we are not making in this linking step? The thing that we're tracking that's not on here. What color is missing? Yeah, exactly. We're not producing ATP in this linking step. That's why it's sometimes included, sometimes not included, because no ATP is made here. So what we end up with at the end in terms of the carbon, right? So we had our 3-carbon pyruvate molecule, we breathed off some CO2, end up with an acetyl group, which we tacked onto coenzyme A, which is derived from vitamin A. And that's what we're sending into the Krebs cycle, is that acetyl-CoA molecule. You can see it's 2 carbons, carbon, carbon. This is not a carbon, this is telling us that's CoA. We didn't try out all the atoms. So the Krebs cycle, citric acid cycle, TCA cycle, is a cycle. So this is going round and round in a loop. And our initial reactants are also going to be our products at the end. That's why it's a loop. That's why it's a cycle. So the Krebs cycle is in the mitochondria. We've moved from the cytosol glycolysis. Pyruvate transported us into the mitochondria, and now we're in the center of the mitochondria in the matrix. Krebs cycle does produce ATP. We have that one ATP coming off per Krebs cycle. So, conceptual question. For each molecule of glucose, how many ATP do you think would be produced by the Krebs cycle? How many times do you think the Krebs cycle is going to run for each molecule of glucose? Yeah, and why is that? Okay.
I think it was the 2 pyruvate.
Yes, exactly. Yeah, we had 2 pyruvates, so everything we talk about after glycolysis happens twice, once for each of those pyruvate molecules. So we get 2 ATP total. We go around this twice. That's not all that much ATP, right? The same as in glycolysis, but nowhere near as much as we're going to produce at the end of this process. So what we really care about for the Krebs cycle is this reduction of the coenzymes. And by coenzymes here, really what we mean is we're making these energized electron carriers. We've seen NADH before. FADH2 is just like another version of an electron carrier. We don't focus that much on it because it just does the same thing as NADH, and it only shows up in the Krebs cycle. That's the main goal of the Krebs cycle, is packing these electrons into NADH so that we can use them in the electron transport chain. So we're feeding in the acetyl-CoA. We're going around, producing lots of NADH, producing some FADH2, giving off some carbon dioxide. So at this point, we'll actually have given off all of the carbon dioxide, all of the carbon we had in that original glucose molecule. We gave off one for each pyruvate in that linking step. Now we're giving 2 carbon dioxides off in each cycle of the Krebs cycle. So that's 3. We double it because we split glucose in half. So it's 6 total, and we have 6 carbons in that original glucose molecule. We're producing our ATP. And we got some water going in. We have some of our initial components going in, and then we just get that. So the stuff on the left-hand side is basically just whatever we need to produce that stuff on the right-hand side. So that's what this is showing us. Conceptually, they've paired it up, not really step by step so much as pairing up these kind of inputs and outputs, the reactants and the products. So again, we are not memorizing all the enzymes. We are not memorizing all the intermediates. But if you want to take a look, it looks like this. Maybe you, I don't know, made a diagram of this in high school or something. People like to memorize it for fun. Nobody ever remembers it in my experience. So that brings us to oxidative phosphorylation. Oh, speaking of, didn't put it up for here, but looking at this, what type of ATP synthesis do you think this is? Those 2 types. Is the same as what we were doing in glycolysis or different? It's a different molecule, but it's still going to be that substrate level phosphorylation. We're still like pulling that phosphate group off of an intermediate. And the reason I remembered to flip back is because we don't get to oxidative phosphorylation until the end of the science step. Part of why sometimes we just call it the oxidative phosphorylation step. So this far, if we're counting our ATP, right, glycolysis gave us 2 net ATP. Right. Our Krebs cycle, each round gave us 1 ATP, but we did it twice. So 1 times 2 equals 2 ATP. Okay, so in total, by the time we get to oxidative phosphorylation, we have produced 4 ATP molecules. 4, nowhere near that 38 number that we saw on Friday, right? We know that overall aerobic cellular respiration is capable of producing a lot more ATP than that. All happening here at the end. So the way that we're going to be doing oxidative phosphorylation, we're going to be moving electrons down the electron transport chain, which is a series of embedded proteins in the inner membrane of the mitochondria. As we move those electrons down, we're basically harvesting their energy, using that energy to produce a gradient of hydrogen ions that we're then going to release and allow to move. And as we allow them to move, we're going to harvest that energy kind of the movement to create our ATP. And we will take a look, but that's fundamentally where we're going here in this oxidative phosphorylation. This is where we need the oxygen. This is the only place we need the oxygen. The oxygen just comes in at the very end, actually. It's going to be our final electron acceptor. That said, while oxidative phosphorylation is the only part where we need the oxygen, the Krebs cycle also is not going to run if we don't have oxygen practically, because the only point of the Krebs cycle is to make those electron carriers. If we can't do the last step, everything gets backed up, and we'll just do glycolysis out in the cytosol if we don't have oxygen. Oxidative phosphorylation requires oxygen, but in low oxygen environments, we're really just going to do glycolysis. So the electron transport chain, the structure that we pair with the process of oxidative phosphorylation. It's a chain of molecules in the inner mitochondrial membrane. It's basically doing these oxidation reduction reactions. What it's doing is pulling off those electrons that we've been packing onto our electron carriers. Right? So as they lose electrons, they're oxidized. That is paired with reduction happening in the chain. This is releasing energy by oxidative phosphorylation. Okay? So we're synthesizing ATP by oxidative phosphorylation. The final enzyme, kind of right after the chain, at the end of the chain, is called ATP synthase, because it synthesizes or makes ATP. ACE tells us it's an enzyme. Now, as we pass down the electron transport chain, we do need to kind of offload our final low energy electrons after we go through this step. And they get accepted by oxygen. And that's actually the step at which we will produce water as well. So it's going to produce some water at the end. We wanted to look at the chemical equations for what's happening here, right? So we can see that we're putting in our electron carrier, essentially. You don't have to memorize these equations, but we're putting in the electron carrier. We're peeling off that hydrogen, right? That's giving us electrons. We also happen to put in another hydrogen on that. But again, you don't have to memorize these equations, right? But this is how we're donating the electrons. We're taking our form where we packed on the electrons earlier in the process where we reduced them. Now we're oxidizing them, and losing those electrons. At the end, after we've used up as much energy as we can, we put those electrons and hydrogens onto some oxygen. So if we wanted to draw it out, right, we just draw our many electron carriers, most of which came from the Krebs cycle, from the CPA cycle. We put them into our nice box of oxidative phosphorylation, which would include this concept of the electron transport chain. We'd get their oxidized forms back, and we get some water. These oxidized forms, those were our original precursor molecules, right? That when we were making NADH, we had to start with NAD plus in order to have something to carry that electron. So now that we have NAD plus again, it's going to loop backwards to the beginning of the process. So these would essentially travel out of the mitochondria into the cytosol and feed into glycolysis. That's where they'd be going. So we can almost think of parts of this process as a loop. We usually think about it as a pathway, but lots of things here do get reused, specifically the electron carriers. And this produces a bunch of ATP. Numbers vary depending on what tissue we're in, what age we are, our genetics, what species we are, but a bunch, right? In the ballpark of 30. Right? So here we have 28, but we can ballpark it around 30. We want to take a look at what this would look like in a picture, right? So our electron transport chain, these embedded molecules. So we have our embedded molecules, and we're not memorizing them either, but they're embedded here in the membrane. The membrane we're talking about is this inner foldy bit of the mitochondria. So this is the inner membrane forming the cristae. Right. So we have our foldy bit, inner membrane. The matrix was in here, this kind of blank space in the center. We have our intermembrane space here. Inter is telling us that it's between something. So what would that intermembrane space be between? If we're looking on the picture. Between the inner membrane and the outer membrane, right? So it's just filling up this area here, spanning this gap. That's our intermembrane space. The reason we're paying attention to that is because what we're doing as we're peeling off these electrons, right, from our electron carriers, we're using these electrons and the proteins in the electron transport chain to pump hydrogens into that space. So we're collecting tons and tons of hydrogens all in here in this intermembrane space. We're going to get to gradients and how they operate in probably this week. If not, we'll get into it next week. But basically, when you have a large concentration of something, it wants to move, right? So if we have a difference in concentration, we'll also talk about differences in charge. Charge plays a role as well, but here we see a difference in concentration of hydrogen ions. So lots in this intermembrane space where I do this blue squiggle, not very many here in the matrix, which is right on the inside. Right, so our matrix would be here, surrounded by the inner membrane. So this concentration gradient is building up what I almost think of as like potential energy. Don't memorize that if that doesn't help you. But we're building up, storing energy kind of in this form of a big gradient because it wants to move, but we don't let it until it's built up high enough. Okay, so when it builds up high enough, when we reach the end of the chain, we bind on to oxygen and we allow the I don't think that was me. We allow the hydrogen ions to flow in. But after this process, we would see equal concentrations of hydrogen ions on either side. They'd reach equilibrium, and then we start to build them up again as we do more aerobic respiration. Before we move forward, I think we have some more pictures like this moving forward. Does anybody want to look at one of these steps again, like right now? We'll come back and touch back on these more in the future too, just to thoroughly cement this. But hopefully this is, yeah, go for it.
Where's number 2 of those intermembrane? It goes 1, 2, 4.
Yeah, I see that. I have that question as well. I think it's just not drawn on.
Okay.
Or, yeah, I think that's right. Yep. Sorry, there's another thing. We're going to see weird numbers where they numbered things in order that they discovered them rather than in order that we use them. But I think that's blood clotting. It's not the electron transport chain. Yeah, this is just showing us more zoomed in concentrations of those hydrogen ions across that inner membrane, across that mitochondrial inner membrane. Right. And as we'll see as we start to talk in depth about membranes, right, hydrogen ions have a positive charge, which means that they're not going to be able to cross the membrane because only non-polar things can cross unless they're allowed to go through a protein. So that's why they don't just kind of dissolve across. If you think hydrogen ions, seems like it would be small, right? But that positive charge means they have to go through a protein in order to get across. Flipping them up, great. So here we have our NADH coming in. We're peeling off those electrons, shooting some hydrogens across into the intermembrane space, travel through our embedded protein, eventually use oxygen to tack on our last set of electrons and some hydrogens, get water, and then we allow our hydrogens to flow across. ATP synthase, I almost think of it as like a revolving door. It spins around, and as it spins, kind of like a windmill, it produces ATP. So our whole process looks something like this. So if we're just tracking our named molecules that we care about that contain carbon, which I just inside, right, we start with our glucose. We make 2-pyruvate, bind it onto some vitamin A. We make this acetyl-CoA. At this point, all of those carbons get lost to carbon dioxide, so we're not tracking carbon-containing molecules anymore. Other thing that we track is ATP, right? So we make 2 ATP in glycolysis, make 2 ATP in the Krebs cycle, and make a whole ton of it in oxidative phosphorylation, adding us up to 32, 34, theoretically, potentially as high as 38. And other thing we note is that we have NAD plus going in and getting reduced, forming NADH, and we see that happening all over the place, right? So we see that in glycolysis. We see it in this linking step with pyruvate, but the place we really see it, see lots of it, so this is what we care about most here, is in the Krebs cycle, which then is used in that oxidative phosphorylation step by the electron transport chain in order to produce ATP and giving us those NAD plus back. So we could send like 2 of them back up to glycolysis. We could send some of them to that linking step. We could send them back to the Krebs cycle and reuse them. Questions about aerobic cellular respiration, this process? Let me just trace these. So that gets us to our overall reaction. Glucose, with some oxygen, some ADP because they're precursors to ATP, some phosphate ions because we're tacking them on to get ATP. We have a variable range. So that's not 32 minus 38, that's 32 to 38. Right? Yeah. I have a reminder in there for myself to pause. So again, I'm not going to ask you to write out the equation on the exam, but you kind of should know what's happening in these steps and the kind of bird's eye view. So that brings us to glucose metabolism in the absence of oxygen, so catabolism, breaking stuff down. Oh, and I did go to the chemistry department and ask about that question you guys had about the link between cations and catabolism and anions and anabolism. And the answer was that when you break something down, you could think of like pulling electrons off of it, and a cation is substantially smaller than an anion. So it has to do with that smallness. So glucose catabolism, glucose breakdown without oxygen. You're in an environment where there's low oxygen availability. And usually when we're thinking humans, right, we're really thinking that you're using up oxygen really fast, right? You're using it up faster than you can breathe it in. We are still able to produce ATP, but we can't use our normal process because that electron transport chain backs up. So it has to wait until there's oxygen available to be the final electron acceptor before we can release that hydrogen gradient. Because we back up all the way up, so the Krebs cycle also stops, because there's no point of doing the Krebs cycle if you can't feed those NADH into the electron transport. However, glycolysis can continue, but we can't do glycolysis if we don't have any NAD plus. Right? So glycolysis isn't the main place we make NADH, but it is a key part of the process. And glycolysis like breaks if we can't feed in any NAD plus. So the NADH that we're producing at the end of glycolysis has to get oxidized. We have to lose that H. We have to lose that electron in order to do glycolysis again. Otherwise, we run out of NAD plus. We can't do this anymore. So that is why we synthesize lactate. So you've heard of lactic acid, right? Potentially building up in your muscles. What we're doing is we're making lactate as a different way of getting that NAD plus. We can kind of convert our pyruvate into it and use that reaction to get our NAD plus back, to use up this electron carrier. So that's the process of lactic acid fermentation.
Why does lactic acid make it muscle sore? Like after you, what's that?
Theory is because it's acidic. I have seen some articles like questioning whether that's really what's happening. But you know how, if you stuck your arm in a vat of acid, right? Like it would burn, and if you spill acid on yourself, then it's really, it's not good. You're supposed to rinse it off. So it's that kind of physical acidic feeling, the idea. So the way that this works overall, right? So if you sit and think about it, it hopefully gets a little bit easier, right? We're just trying to do glycolysis to make ATP, right? This is what we want. This is our goal. We want this ATP, right? So we have to do the side process just in order to get that NAD plus back. So this process of producing lactate from pyruvate, the lactate dehydrogenase is the enzyme that does this. As we make lactate, one of the steps of making lactate is oxidizing that NADH. So that's the whole reason we're doing this. We don't really care about having a bunch of lactate. Making the lactate is not the goal. Being able to continue glycolysis is the goal. This is just a boring textbook video, and we are at time. So next time we'll do a little case study, and we'll talk a little bit about other carbohydrates, lipids, and proteins that can